Children, today I am going to take the second part of genetic disorders that are coming under the category Mendelian disorders. Same chapter, Principles of Inheritance. So, recap of Mendelian disorders. Mendelian disorders gets its name first of all because it follows the Mendel's laws of inheritance. Then it is caused due to mutation of a gene or point mutation, and it is studied by a pedigree chart. We are going to learn 5 Mendelian disorders, and I have clubbed them in 3 categories. The first 2 is hemophilia and color blindness. They are coming under sex-linked recessive inheritance. So, the first 2, hemophilia and color blindness, are coming under sex-linked inheritance. Then we are having the next 2 disorders, sickle cell anemia and thalassemia. See, thalassemia and sickle cell anemia coming under autosome linked inheritance. And lastly, we are having phenylketonuria, which is coming under an inborn metabolic disorder that is also autosomal recessive linked inheritance. So, the 5 types of disorders, the 5 disorders coming under Mendelian pattern. So, let us get started. So, first I am going to deal with hemophilia, and I am going to emphasize these points. 1, the pattern of inheritance, 2, the cause, 3, the symptoms, and 4, the condition. The pattern of inheritance I have already mentioned, it is a sex-linked recessive inheritance. Then, the cause of hemophilia, as we all know, Mendelian disorders are caused due to mutation. So, the cause again we have to mention, it is caused due to mutation, and the mutation, which mutation? Point mutation. And now the symptoms. Symptoms of hemophilia. You are familiar with hemophilia, it is referred to as a royal disease. Why it is referred to as the royal disease? Because it is present in the royal family of England. Queen Victoria is the carrier of this disease, and many of her descendants are suffering from hemophilia. Now, coming to this condition, what is hemophilia? The person who is having hemophilia, the blood is not able to clot. Even a small bruise or cut will lead to excessive bleeding. Okay, it becomes so severe when it can be very fatal. So, what is hemophilia? It's a condition where the blood fails to clot. In normal individuals who are normal, if a small clot, if there's a small cut, after some time it gets clotted, it gets sealed, it does not bleed. But these people, they lack, the blood has the inability to clot, and therefore a minor cut also leads to excessive bleeding. So, that is called hemophilia. That condition is called hemophilia. Now, coming to color blindness. Color blindness also, everything is the same. Pattern, I am repeating, pattern of inheritance, sex-linked inheritance. The cause is point mutation. Third, the symptoms here is, as the name suggests, the person is unable to distinguish between red and green colors. There are different types of color blindness. Red and green, the basic or the primary colors. But in your text it is mentioned the inability to distinguish between red and green colors. And now I have shown the Ishihara's plate. It's a detection for color blindness. See, this is the Ishihara's plate, which is used to identify color blindness. Now, if the person, see, you also look at this plate. Are you able to read which number can you identify from this pattern template? If you can identify the number 16, you are not colorblind. Now, in this, what can you identify? If it's 73, you are not colorblind. But colorblind people will not read 16 or 73. They will read it as some other number, or they cannot identify these numbers from this Ishihara's template. Clear? So, this is a test to identify whether the person is red-green colorblind. I told you there are different types of color blindness, but here in the text they have emphasized the inability to distinguish between red and green colors. So, once again, hemophilia and colorblind giving common characteristics. Up till number 3 it is common to both the diseases. The pattern of inheritance of hemophilia and colorblind is sex-linked recessive inheritance. Second, the cause of hemophilia or colorblind is mutation. And third, the symptoms for hemophilia is the failure to clot. The blood lacks the failure to clot, whereas colorblind is the inability to distinguish between red and green colors. Now the genotypes. Fourth, genotype is also common to both. Now, to work out problems in hemophilia and colorblind, we have to know the genotypes of the individuals, both male and females. Now I am going to give the genotypes of the males. That's the basic idea. Males are XY and female is XX. And I told you hemophilia and colorblind is sex-linked inheritance. So it's linked with the sex chromosome. Which is the sex chromosome it's linked to? Remember, it's linked to the X chromosome. Now, I'm using another color to highlight this. Let capital C in the case of Helia means normal clotting. So let the alphabet capital C determines normal clotting, whereas small c hemophilia, that is non-clotting. Okay, so let capital C determine normal clotting and small c hemophilia. So what will be the possible genotypes for the females? Ready? So this is the female. I am going to give all the possible genotypes for the female. Ready? And we are putting it is linked to the X chromosome, and we are placing the C's at the superscript. So we have X capital C, X capital C, then we are having X capital C, X small c, and we are having one more X small c, X small c. See, look at the possible genotypes in females. X capital C, X capital C. So what is the phenotype of this individual? Is this individual normal or hemophilic? Normal. So capital C means it's a normal individual, normal female. Then X capital C, X small c, again she is also normal, but remember she is a carrier. So I have indicated with C. But look at the last category, X small c, X small c, C small c means hemophilic. So this is definitely hemophilic, the defective female. So these are the three possible genotypes coming under females. Now males, it's linked with the X chromosome. So we have X capital C, Y and X small c, Y. So only two options are there. We can't go for the third option because it's only linked with the X chromosome. So we are having two genotypes, X capital C, Y, X small c, Y. What does this indicate? This is a normal male and this is a hemophilic male. So there are two genotypes coming under males and three genotypes coming under females. Clear? So once again explaining the genotype. In the case of hemophilia, let capital C be normal clotting and small c be hemophilic. So the possible genotypes for females are three genotypes. One, X capital C, which is a normal female. Second, X capital C, X small c, which is a carrier female. She is also normal. And third, X small c, she is hemophilic. Okay, now the possible genotypes in males. Only two genotypes are possible in males. One, X capital C, Y, he is a normal male. And two, X small c, Y, he is a hemophilic male. Okay, so how many genotypes are possible in humans? There are 5 types, 3 in females and 2 in males. And remember, they are linked with the X chromosome. So this I explained from the point of view of hemophilia. Now the same pattern of inheritance is used for color blindness. So now what I am going to do is just changing what capital C and small c stands for. Now here capital C stands for normal color vision and small c stands for color blind. See, I just changed the indication. and the remaining all stands the same. So now again I am explaining from the point of view of color blind. Let capital C indicate color vision, small c indicate color blindness. So the possible genotypes coming for females are 3 types: X capital C normal, X capital C, X small c carrier but normal, X small c, she is color blind. Okay, just rewording it. Now coming in the case of males, there are only two options. X capital C Y, it's a normal vision male, and X small c Y is a color blind male. Clear? The same pattern of inheritance. Only thing you have to reword what capital C and small c indicates. Clear? Now, one very common question here that is asked is, rarely females are colorblind. Why females are rarely colorblind, or why females are rarely hemophilic? Why? If they have to be, see, if the female has to be colorblind or hemophilic, definitely the father should be having the drawback. See, the father should have the drawback and the mother at least should be a carrier. This is the condition. For a female to be colorblind or hemophilic, see, this is a female. If she has to get small c, one she has to get it from the father, the other small c she has to get it from the mother. So definitely what should the father be? The father should be having the defect. If it's colorblind, the father should be colorblind. If it's hemophilia, the father should be hemophiliac. And the mother should be a carrier. Definitely the mother should be a carrier bearing X capital C, X small c. So this female gets this one from the father, X small c from the father, and this X small c from the mother. So definitely for the female it should be double recessive condition. So she gets the X one from the father and the other X from the mother. So the X she gets from the father, X small c. So that father, definitely the other option we can't go for. Normal X capital C, then she gets X capital C. So she should get X small c, means the father should have the defect, and definitely the mother should be either colorblind or should be at least a carrier. Therefore, it's very rare. Females are rare in the population having the disease, very rarely found in a population. Females, colorblind females and hemophilic females are very rare in the population. This is the reason. Once again, a recap. I clubbed hemophilia and colorblind because the 4 points are common. Not 4 points, the third point only, there's a difference. Once again, we'll go a quick recap. Pattern of inheritance of both hemophilia and colorblind, sex-linked recessive inheritance. See, sex-linked recessive inheritance. Then the cause is mutation because it's coming under Mendelian disorder, so you have to specify that. Then symptoms. For hemophilia, symptoms is failure of the blood to clot. For colorblind is the failure to distinguish between red and green colors. That is the symptoms. And lastly, the genotypes. Genotypes you have to specify or indicate what C stands for. I have already explained what capital C and what small c. Remember, capital C stands for normal, whether it's normal vision or color vision or normal clotting, and small c stands for the drawback. And therefore, you give the genotypes of the females. There are 3 genotypes and genotypes of the males. Okay, so coming under this category of disease, the 2 which I clubbed, hemophilia and colorblindness, learn it under these headings: pattern of inheritance, the cause, symptoms and genotypes. I hope this is clear. I have emphasized 4 points for these 2 diseases. Okay, so we finished the first pair. Next, thalassemia and sickle cell anemia. So 2 diseases I have taken: thalassemia, number 3, and number 4, sickle cell anemia. Now, for these 2 diseases, the first 3 points are common. So, pattern of inheritance. This is the pattern of inheritance. What is the pattern of inheritance? Autosome linked recessive inheritance. It is an autosome linked recessive. So, the pattern is from the autosome, not from the sex chromosome. Autosome linked recessive inheritance. That's a pattern of inheritance for both thalassemia and sickle cell anemia. Now, the cause. We know the cause, mutation, because it's coming under Mendelian disorders. The cause is mutation. Now, the symptoms. For both of the diseases, the symptoms is the same. What is the symptoms? Anemia. We are familiar with dietary anemia, lack of iron in our diet. We become tired and we take a lot of iron tonics. That's only dietary anemia, getting tired easily because the oxygen carrying capacity is low. Now here also the symptoms are same, very chronic, but anemia. The symptom is just anemia. Why anemia? Because which molecule carries oxygen in our blood? It is the hemoglobin. So this oxygen carrying capacity of hemoglobin decreases and therefore the person gets tired easily and therefore he gets a disease called anemia. Now we are going to analyze the condition. In both the cases, the RBCs are distorted in shape. The shape is distorted. What is the actual shape of RBC? It is disc and biconcave. So you are familiar with the shape of RBC. It is disc biconcave shape, disc-like and biconcave because it lacks the nucleus. Okay, that's a normal shape. This is normal. But under these two conditions, what happens is that they become distorted. See, I've highlighted the shape of the RBC gets distorted. In sickle cell anemia, look at sickle cell anemia. So look at sickle cell anemia, look at the shape. This is a normal RBC. See, normal RBC. But look what has happened to the shape of the RBC. It has become the shape of a sickle. Now look at thalassemia. This is a normal RBC, but look at the shape. Look, disfigured. It has become disfigured. In both the cases, the RBC gets distorted. Okay. Now, the reason is different. But so once again, the pattern of inheritance, autosome link recessive inheritance. The cause is mutation. Symptoms is anemia. Anemia is lack of oxygen carrying capacity and therefore the person gets easily tired. And lastly, the condition is both the cases, the RBC, the shape of the RBC is distorted. In sickle cell anemia, it's sickle shape. In the thalassemia also, it's distorted. Okay, the reason is because of a defective hemoglobin. Because the hemoglobin has become distorted or defective, the oxygen carrying capacity of hemoglobin also has decreased. Now we are going to analyze both. The difference comes here. Okay, I'm going to bifurcate it to show the difference. One is a qualitative problem. Qualitative. And the other is a quantitative problem. So which is a qualitative problem? Qualitative problem is sickle cell anemia. Sickle cell anemia is a qualitative problem and quantitative problem is thalassemia. So let's analyze these two. Now let's deal with sickle cell anemia. See? The same text figure I have pulled out. So, in sickle cell anemia, what has happened? Before going into the details of sickle cell anemia, let's learn the structure of a normal hemoglobin. And from that, we will elicit the drawback. See, the description of a normal hemoglobin. A hemoglobin molecule is a tetramer consisting of two pairs of similar polypeptide chains called globin chains. So, hemoglobin is a tetramer consisting of 4 globin chains, namely 2 alpha globins and 2 beta globins. And these alpha and beta globins are connected to heme. These 4 chains are attached to heme, which is a complex of iron. So, I am going to show you in this figure. See, here is the beta, one beta chain and this is the second beta chain. So what is in yellow is the beta chain and what is in red is the alpha chains. See, and you can see the heme units given as blue disc. Okay, so once again, hemoglobin comprises 4 globin units or 4 globin chains. 2 alphas and 2 betas. The alpha is called alpha 1, alpha 2, and the betas are called beta 1, beta 2. See beta 1, beta 2, which is in yellow, and alpha 1 and alpha 2. And all these 4 chains, how are they connected? They are connected to heme of iron. A heme complex is there. See what I've highlighted in yellow. I'm removing all the highlighted portions and see the real structure. Okay, so this is a tetramer of hemoglobin. Now we're going to see the drawbacks. So you understood, each hemoglobin, if it is normal, it should have 2 alpha, 2 beta connected to the heme unit. There are 4 heme units, see blue color, heme units, which is forming a part of hemoglobin. Clear? Now we are going to see sickle cell anemia. Now I told you 2 alpha, 2 beta, and I told you sickle cell anemia is coming under qualitative problem or qualitative disorder. What has happened is, look at this chain, which is indicated 1, 2, 3, 4, 5, 6 with a number of circles. Each circle indicates amino acids. See, look at, there are 7 amino acids. Okay, again I'll make it bigger. See, there are 7 amino acids. Later we will be familiar, in the next chapter we will be learning with almost 20 of the amino acids. So by that time you will be familiar with these amino acids. But by now you know some of the amino acids because we have learned in chapter 10 biomolecules in 11th standard. We are familiar with some of the amino acids. See, look at the amino acids that forms the beta globin chain. See, remember the beta globin chain of hemoglobin. The amino acid 1 is valine, 2 is histidine, 3 is leucine, 4 threonine, 5 proline, 6 glutamic acid and 7 glutamic acid. All what you need to learn is only the 6th amino acid. So the 6th amino acid or the amino acid in the 6th position of the beta globin chain. So once again, the amino acid in the 6th position of the beta globin chain of hemoglobin. So only this is what you need to learn from this picture. So the 6th position should be glutamic acid, but what has happened, instead of glutamic acid, it's replaced by another amino acid. Look, this is the drawback. It's replaced by another amino acid called valine. So normally it should be glutamic acid in the 6th position, but what has happened, due to mutation, glutamic acid is replaced by valine. So this is a defect which can be categorized as a qualitative defect. The quality of the beta chain has altered. So once again, a normal beta chain in the 6th position should have glutamic acid. But here what has happened, due to mutation, the glutamic acid is replaced by a wrong amino acid called valine. As a result, what type of RBCs are produced? Because a wrong amino acid, instead of glutamic acid, valine is replacing in the glutamic acid in the 6th position of the beta globin chain of hemoglobin. What type of RBC is synthesized? A defective RBC, a distorted RBC, an RBC which is having sickle shaped. Whereas here, if it was glutamic acid, what would have been the resultant RBC produced? It would have been the normal RBC, which is disc biconcave shape and which is able to carry oxygen. So only you emphasize on the 6th position, need not learn the other amino acids as such. So once again, what is sickle cell anemia? What is the condition? It's a qualitative problem wherein the problem is in the beta globin chain. We told there are 2 alpha, 2 betas. Here the problem is, the defect is in the beta chain. The 6th position of the beta chain should be, in normal case, it should be glutamic acid. Then it will result in the normal RBC, which is disc shaped. But what has happened, due to mutation in the 6th position, instead of glutamic acid, valine, a wrong amino acid, got substituted, and as a result, what type of RBC is produced? A distorted RBC which is having sickle shape and therefore inability to carry oxygen. Clear? I hope you have understood sickle cell anemia. It's a qualitative process. It's a qualitative problem. Now let's go to the other, thalassemia. Thalassemia is a quantitative problem. Now quantitative, from that you get a clue, the number. Okay, I told you in a normal hemoglobin there are alpha chains and beta chains. And how many alphas are there? 2 alpha, alpha 1, alpha 2. Beta, how many are there? Beta 1, beta 2. Okay, so this is the normal hemoglobin, but sometimes the rate of synthesis of either alpha or beta decreases. also, due to mutation, the rate of synthesis. So the drawback is this: rate of synthesis of one of the change, globin change, either alpha or beta decreases, not increases, decreases. Okay, so it's a quantitative problem. Why? There should be 2 alphas to 2 betas, but sometimes what happens, it will be 1 alpha to 2 betas, or sometimes it will be 1 beta to 2 alphas. Actually, it should be equal. The rate of synthesis of both alpha and beta should be equal. But what happens? The synthesis of either one, alpha or beta, decreases. So it becomes a quantitative disorder resulting in thalassemia. So accordingly, there are 2 types of thalassemia. The 2 types of thalassemia hence will be called, it's easy to remember, alpha thalassemia and beta thalassemia. Now, the first difference between alpha and beta thalassemia, as I already indicated, the first difference is rate of synthesis. In alpha thalassemia, the rate of synthesis of alpha is low. The rate of synthesis of alpha chain is low. Whereas in beta thalassemia, the rate of synthesis of beta chain is low. So I have highlighted low. Okay, in normal person, both are equal. Alpha and beta are synthesized equally. But in alpha thalassemia, alpha is low, but beta is normal. On beta thalassemia, the number of beta is low compared to alpha. Alpha is normal. So you put it like this way: the rate of synthesis of alpha chain is low or decrease in alpha thalassemia. In beta thalassemia, the rate of synthesis of beta globin chain is decreased. Clear? That is one difference. Second difference is alpha thalassemia is controlled by 2 closely linked genes. Alpha thalassemia is controlled by 2 closely linked genes. The name of those 2 genes are indicated as HBA1 and HBA2. So alpha thalassemia is controlled by 2 closely linked genes, namely HBA1 and HBA2, located on chromosome number 16, and not of one parent, of both the parents. The father and mother should have this drawback, and when they marry, the child that is formed will have thalassemia. If one parent has this mutation in these 2 genes and the other is normal, then the child born will not get thalassemia. But in this case, what happens? Both the parents, each of the parents, specify in both the parents, on each of the parents, HBA1 and HBA2 gene, closely linked gene on chromosome 16 should have the defect or should have the mutation. Okay, that's the second difference. Now here in beta thalassemia, the name of the gene is HBB. HBB, remember to write it in caps lock. And HBB gene located on chromosome 11 of both the parents. Both the parents should have the drawback, and if they happen to marry, the child born will have beta thalassemia. So once again, the difference between beta thalassemia and alpha thalassemia. Alpha thalassemia, the rate of synthesis of alpha globin chain is low. Beta thalassemia, the rate of synthesis of beta globin chain is low. Second difference, alpha thalassemia is controlled by 2 genes, HBA1 and HBA2, located on chromosome 16 of each parent, whereas beta thalassemia is controlled by the gene HBB, located on chromosome 11 of both the parents. Emphasize both the parents, and hence we can say thalassemia is a quantitative problem and sickle cell anemia is a qualitative problem. Now taking a quick recap of autosome linked recessive disorder. First, common to both. I told you I clubbed the diseases together because of many common points. So thalassemia and sickle cell anemia, pattern of inheritance, autosome linked recessive inheritance. Second, so I am taking it. Second, the cause of disease, we know it's coming under Mendelian disorder, so it's caused due to mutation. Then symptoms. Symptoms in both the cases, it's anemia, that is getting tired easily because the oxygen carrying capacity of the hemoglobin is low. And why the oxygen carrying capacity of hemoglobin is low? Because the hemoglobin is a defective hemoglobin. There might be a qualitative defect or a quantitative defect. So accordingly, there are 2 types of disorders. If it is a qualitative defect, it is sickle cell anemia, and if it's a quantitative defect, it's thalassemia. Now, getting to sickle cell anemia, what is a qualitative defect? What's a qualitative defect? The sixth position of the beta globin chain should be glutamic acid. But here, instead of glutamic acid, a wrong amino acid is substituted, valine. Hence, the RBCs that are produced as a result of this will be defective. The distorted shape of that is sickle shape. Whereas in quantitative, that is thalassemia, which is indicating a quantitative process, we know that hemoglobin is a tetramer consisting of 2 alpha and 2 beta, but thalassemia results when one of the globin chains synthesis are decreased. In the case of, and we have two types, in the case of alpha thalassemia, there is a decreased rate of synthesis of alpha, and it's controlled by two genes, HbA1 and HbA2, located on chromosome 16 of both the parents. Now, beta thalassemia, the cause is due to decreased rate of synthesis of beta globin chain, and this defect is controlled by the gene HBB, located on chromosome 11 of both the parents. So, I hope you have understood the two types of thalassemia, alpha and beta, coming under thalassemia, and both thalassemia and sickle cell anemia are coming under autosome linked recessive inheritance. Clear? Now, problems are only coming from sickle cell anemia, and hence warrants the genotypes for sickle cell anemia, the genotypes of sickle cell anemia. And the most easiest problem in genetics is related to sickle cell anemia. There are only 3 types of genotypes, irrespective of the sex. I told you it's autosome linked, so there's no importance for the sex chromosome. The genotypes are H, I'm writing HBB showing the hemoglobin. Okay, and because we are diploid, I'm showing HBB twice. Now, in the first case, HBA, superscript HBA. Second case, HBA, superscript A and HBS. Then HBS. Now, I gave the genotypes. Look at the first genotype, HBA. This is normal person, irrespective of the sex, normal person. You not say normal male or normal female, it's a normal person. Next case is HBA-HBS. HBA-HBS. This is a carrier person. And the third category we have HBS-HBS. This person is sickle cell anemic. So, the 3 possible genotypes are HBA-HBA, normal person, HBA-HBS, a carrier person, he or she is also normal, and last, which is a defective or sickle cell anemic person, is HBS-HBS. This is the most easy. You get problems based on sickle cell anemia, it's very easy, because the only options are between these 3 genotypes, irrespective of the sex. So, only problems are coming from sickle cell anemia, and remember the genotypes. Clear? The 3 genotypes, HBA-HBA indicating normal person, HBA-HBS, it's also normal but carrier, and HBS-HBS is also coming, and the third genotype, but that person is sickle cell anemia. The last disorder is phenylketonuria. We have learned phenylketonuria. You are familiar with this picture. The same picture was given in the previous video. What does this indicate? PKU. And what we have learned is one gene controls more than one character, and which is that gene, pleiotropic gene. This pleiotropic gene controls 3 characters: mental retardation, lack of pigmentation of the skin, and lack of pigmentation of the hair. So that is with context to pattern of inheritance called pleiotropy. Now again we are learning the same disorder, phenylketonuria, which is coming under the pattern of inheritance. Pattern of inheritance is, it is also autosome linked recessive disorder, but it is inborn metabolic disorder. So it is coming under, you have to complete the statement writing both it is a metabolic disorder as well as it is autosome linked recessive inheritance. Clear? Then, now condition. What is the cause of the condition? We know the cause is definitely mutation. We know mutation because it's coming under Mendelian disorder, definitely it is mutation. And now we can see the condition. In normal person, phenylalanine gets converted to tyrosine with the help of the enzyme phenylalanine hydroxylase. once again, in a normal person, this enzyme phenylalanine is present. Phenylalanine hydroxylase is present, and the function of this enzyme is to convert phenylalanine into tyrosine. Okay, but in the phenylketonureacs, in this defect, what happens? This enzyme is absent due to mutation. Due to mutation, this enzyme is not present. It is lacking. Phenylalanine hydroxylase is absent. So, will this conversion take place? Phenylalanine to tyrosine? It does not take place. What happens instead is, this phenylalanine gets converted into phenyl pyruvic acid and its derivatives. And what happens is this phenyl pyruvic acid and the derivatives get accumulated in the head, okay, in the cerebrospinal fluid, and therefore does not allow the growth of the development of the brain, and such children are mentally retarded, get accumulated in the cerebrospinal fluid. And not only that, a lot of these phenyl pyruvic acid is also excreted out through the urine because of the poor absorption capability of the kidneys. So the two symptoms is mental retardation. Why mental retardation? Because of the accumulation of phenyl pyruvic acid and its derivatives in the head. Therefore, the brain does not develop, they become mentally retarded, and a lot of phenyl pyruvic acid and its derivatives are lost through the urine, are excreted through the urine, because the kidneys are very poor, does not function properly, they do not absorb this particular derivative. Clear? So the normal condition, in normal individual, what is present? Phenylalanine hydroxylase. This is an enzyme which converts phenylalanine into tyrosine. But in this drawback, what's the drawback? Phenylketonuria. What happens is, this particular enzyme is absent. So this conversion does not take place. Instead, this phenylalanine gets converted into phenylalanine pyruvic acid and its derivatives, and it gets accumulated in the brain or in the head, as well as a lot is excreted through the urine because of the poor absorption capacity of the kidney. Clear? I hope you have understood. So under phenylketonuria, the pattern of inheritance is autosomal recessive inheritance, which is an inborn metabolic disorder. Then the cause, I said you, mutation, and third, the condition is lacking. These children or these individuals lack the enzyme phenylalanine hydroxylase, which is essential for the conversion of phenylalanine to tyrosine. Instead, what happens is, phenylalanine gets converted into phenyl pyruvic acid and its derivatives, which gets accumulated in the head and excreted through the urine. Clear? I hope these 5 disorders are clear, coming under Mendelian disorders. Learn it in the same way, because compare and contrast is asked like this. It's compare and contrast between hemophilia and color blindness, compare and contrast between sickle cell anemia and thalassemia, and phenylketonuria also comes separately. Okay, so I hope you have understood Mendelian disorders. Thank you.